today we begin this learning session by oncodiagnostic group, and it was important to us to have an expert in this domain. So I have the pleasure and the honor to introduce Daniëlle Vugts, an associate professor in radiology and nuclear medicine in VUMC Hospital in Amsterdam. Daniëlle is very implicated and involved in dynamic in a lot of domains like neurodegenerative disease and oncodiagnostic area with more than almost 80 publication reference. She's one of the women who have the better knowledge of immunopet radiolabeled with Zirconium 89, and her team and her begin the first clinical studies in human with Zirconium 89 antibodies. So SoFRa and me are very honored to have someone like you sharing your expertise and knowledge of immunopet radiolabeling with Zirconium 89 with us. So Daniëlle, it's your turn.
Yeah, thank you very much. I will share my presentation. Wait a second. First, close some things. I'm not sure what you see right now. Is it the presentation or is it the that you see two slides?
We see two slides.
Oh yeah. I never know how to solve this. Wait a second. That's what you get when you have two screens. Yes. And I'm not very used to that.
Yes.
Is it now better?
Yes, yes.
Okay, great. Okay, thank you very much. And indeed, in the next half an hour, I will try to explain a lot about zirconium. And if you have questions, please feel free to ask. And if you want to have information later on, also you're welcome to send me an email about that. So a little bit, I will tell a little bit about the background, why we started all this work, and then some, the radiochemistry behind zirconium, the clinical examples, and some newest developments. So first, some background about antibodies and drug development. And what you can see is that in the last years, there's really a shift towards biologicals being the most sold, but also being the most developed. And here are some figures and also some information about antibodies. And what you can see is that 7 of the top 10 drug blockbusters were biologicals in 2019. And they represent 65 billion out of the 87 billion. And just to put the radiopharmacy into perspective, is that one top 10 radiopharmaceutical values the whole radiopharmaceutical market. But I think there's a lot to gain, especially in drug development with imaging. So a little bit more about antibodies. By the end of 2020, I think there were 109 antibodies approved by the FDA and/or the EMA, of which 43 were for cancer, but there are a lot in clinical trials right now. So over 550 antibody therapeutics, of which 79 in late-stage clinical trials. And this paper, Antibodies to Watch, I think it's already published for years. And every year there's in the journal MOPS, there's this paper about antibodies to watch, where you can find very useful information about the antibody development domain. And what is anticipated that by 2024, biologicals will contribute 50% of the top 100 product sales. But these antibodies are very expensive, as you probably all know, and they're also not very efficient for every patient. And that's where we were questioning ourselves, is there a role for academic molecular imaging? So if I talk about antibodies, so let's say 15 years ago, that were the standard IgGs, so rituximab, cetuximab, trastuzumab, starting with chimeric antibodies and later on humanized and human antibodies. But it's not just antibodies anymore, it's antibody drug conjugates, so an antibody equipped with a small, very toxic drug. Immune checkpoint inhibitors, so not targeting the tumor cell, but neighboring immune cells like T cells, but also fusion antibodies by multispecific antibodies, antibody fragments, antibody-like scaffolds, but also oligonucleotides, nanoparticles, and even cells. And that's where we think that imaging can help to better understand these molecules. So if you look at as I already just mentioned, pharmaceutical drug development is not very efficient. So only 10% of the drugs that will enter clinical development will reach the market. So a lot of effort is put in early phase, but also later phase clinical trials, and these drugs still fail. Furthermore, here on the right, you can see the effectiveness of drugs. So for example, in the case of cancer, I think it's almost the worst scenario you can have. Only 25% of the patients will benefit from treatment. Furthermore, because of this low success rate, the drug development process is very expensive, and typically timelines are around 10 to 15 years. And only in the late phase clinical trials, added value is demonstrated, which means that a lot of money is spent on drugs which fail in the later stage. So if you look at the worldwide sales is around 1,000 billion. And let's say if you look at all the different diseases, for 600 billion, this is spent in vain. So we think that with imaging, we could hopefully improve this. And that means do imaging studies in early phase clinical trials. We think that the biology and the targets must be understood much better. And also the drug design and development should become more efficient and cheaper. So selection of highly potent drugs at the earliest stage with less patients in trials, and then also showing the distinguished properties of a drug. For example, I think for the PD-1, PD-L1 axis, there are a lot of antibodies developed, several approved. So they're all targeting the same domain. Like, do we need more of that? I don't know. And furthermore, with imaging, we hope that drug treatment should also become more personalized and more precise. So if we can image a drug, what of the following points can we get information on? So if you can label a drug, you can look at the pharmacokinetics and biodistribution. Typically for a phase 1 trial, it's only looking at, is this tolerated by a patient? But there's nothing known about, does the drug hit the target? Furthermore, you can look at confirmation of disease targeting and also anticipate on toxicity. And that's also connected to the next point. Are there any sinks? I will have a nice example about that later. So location of a certain receptor or antigen on healthy tissue. This typically needs to be saturated before you can get tumor targeting. And also having this information on a patient level, you can also think about probably your overdosing if you give more. And by giving the optimal dose based on targeting, you could probably also prevent side effects. And we have the idea we think that with imaging, you can also show the distinguished properties of a drug. And for this, you need a labeled drug. And we think this is more informative than using, for example, FDG, so really response monitoring. And you need an antibody, of course, for that. But I'll come back to that later. So when we started this work, already quite some years ago, even before I started in a group here, we had some requirements we set for ourselves. So since especially the larger pharmaceutical and also smaller pharmaceutical companies are developing the new drugs, the method that we apply in radiochemistry should be acceptable for the pharma industry, but also to the regulatory instances. That means that the first thing that we set to ourselves, the labeling may not affect the properties of the drug. So it should be an inert radio labeling. Furthermore, we should develop to a stage with chemicals, reagents, materials that are acceptable in production of tracers for human use. So we should comply to GMP, good manufacturing practice. Probably all familiar with that. Furthermore, we should not be only able to do this in Amsterdam, but also in other sites. So we want and help others to also set this up. And it should be easy to disseminate, broadly applicable, and also apply it in a standardized way. Furthermore, the first clinical study we did, we could do without any additional tox studies of the radio-labeled antibody versus the cold antibody, which was anyway evaluated in toxicology studies. This set, of course, the stage for next applications. By showing that you do an inert labeling, we didn't have to do any tox studies. Furthermore, we always hoped that imaging does not delay the initiation of clinical trials. That's not always easy, especially in collaboration with pharmaceutical companies who are not used yet with doing radio tracers. So you have to educate them, but thereafter it can go quite quickly. We have an example that we started a study together with Roche. We started in January with setting up the radiochemistry, and before the summer it was all validated. I think that was a very fast process. Sometimes it can take longer because the radiochemistry is not working, or some quality controls are difficult to set up. So let's say half a year is very fast, then the IMPD is ready, but it can also take up to a year. Furthermore, it should be cost effective. Thinking about the expensive drugs, that's quite easily achieved, of course. If you can show already in phase 1 clinical trials that a drug will make it or not. Typically, we now call it de-risking drug development, because showing in an earlier phase that the drug will never be effective. But this is already very cost effective, of course. And with regard to the imaging, our colleagues made standardized and validated imaging quantification procedures, like was done with Earl for FDG, the same with Zirconium. Then for those of you who do not know the characteristics of zirconium, so it's a PET isotope and decays for 23% via beta plus to yttrium 89. And it decays first to an isomeric transition that you see here. So like the metastable form like in technetium. And this has a half-life of 15.7 seconds. And then you have a gamma of 900 kilo-electron-volt for about 100%. This decays around 60 seconds later and therefore does not interfere in the PET signal. The energy of the beta plus particle is comparable to carbon-11, slightly higher in energy than fluorine 18, and the resolution is therefore comparable to carbon-11, but slightly worse than fluorine 18, but way better than, for example, gallium-68, which has a much higher beta plus energy. And the production can be done in a cyclotron by bombarding yttrium 89 with protons. Neutron goes out and you get zirconium 89. And the production, you you can do using an yttrium disk in a solid target holder. And if you bombard with protons with lower energy lower than 13 MeV, you get zirconium 89. If you go in with higher energy, you get zirconium 88. So you don't want that. It has a very long half-life. Impurity you do not want. And the zirconium can be separated from the yttrium by dissolving it in 6 molar HCl and then separating it by affinity chromatography. So we make column material which is equipped with hydroxamate groups. The zirconium is trapped on the hydroxamate and the yttrium doesn't have an affinity there and is eluted. And when all the yttrium is eluted, you wash the column and then you can elute your zirconium from the column with 1 molar oxalic acid. And this gives you then the solution that you can nowadays purchase commercially, for example, from Burke and Elmer, but you can also make it yourself, of course. With regard to the chemistry, to date, we have one chelator which is used in clinical trials. Hopefully this will change soon. I will come back to that at the end of my talk. And this chelator is desferrioxamine or DFO or desferral. This chelator is an hexadentate ligand. On the next slide, I have the actual structure. Here's just a graphical representation. And what we have is that this DFO consists of 3 hydroxamate groups. It's an acyclic chelator, and it has an amine for functionalization with linkers that can be attached then to an antibody. We have developed 2 linkers, a succinyl linker that you see here, or an isothiocyanate linker that you see here on the right. We have done several preclinical and clinical studies with both chelators. They behave exactly the same. The only thing is that the second one, if you store the radiolabeled product in saline, we see some instability over time. If you prevent the use of saline, for example, using sodium acetate or a histidine buffer, this can also be kept very stable. And what is furthermore important to mention, that's also something I will come back to at the end, is that in preclinical models, we see a little bit of zirconium uptake in the bone, which is free zirconium. We don't know whether this is a catabolic process, so due to uptake in certain organs and then catabolism in the organ and then some free zirconium which is released. Most probably that is the case, and that's why we developed also a new chelator for zirconium. With regard to the radiochemistry, here you see the chelator zirconium, and this is then the isothiocyanate linker. You can couple that at basic pH, 30 minutes, then you have this complex, and then you can immediately do a zirconium labeling. Of course, purification in between to get rid of the excess of the chelator. And this is all written down in this Nature Protocols, but also this paper from 11 years ago. And with these chelators, all the production of zirconium, scanner calibration, this is all made available over the last years. Then some clinical examples. I want to start with the first clinical trial ever, already published 15 years ago, before I started here in the group. And this is all due to the hard work of Guus van Dongen, my colleague, and he was my previous boss. And this was he was then working at the ENT department, so had a neck cancer. And there they had an antibody targeting CD44V6 who had neck cancer. And it's called U36. And this antibody was labeled with zirconium. And in this study, for example, the dosimetry was determined, and also some tumor targeting, of course. And what was proven also, of course, the safety of using zirconium. So the feasibility and the safety was proven in this first clinical trial, and all primary tumors could be detected with zirconium. an antibody dose of 10 milligram was given, since between 2 and 52 milligrams, there was no antibody dose dependent pharmacokinetics observed. So quite linear. So that's important for your imaging. And the dosimetry is high, we know. It's 0.6 millisievert per megabecquerel. These first trials were done with 74 megabecquerels, and that gives then a really serious dose to the patient. Right now, we give 37 megabecquerels. Because of this results then in around 20 millisievert dose, and then we are allowed right now to dose the patient twice with the tracer. Come back to that also later, because with the total body PET scanners getting on the market, we can lower the dose of the zirconium, but still have good, even better images. I've seen the first scans with zirconium in humans, and that looks really, really good. So right now the dose is still a concern, but with the total body PET, I think, yeah, the interest in zirconium will only increase. And we are in the lucky position to get one early next year. So we really hope that this will bring much better images, because they are still quite noisy with zirconium, and that you also can detect better between noise and a real signal, of course. And the dose is lower for the patient, which is absolutely most important. So this is then, the first one was the CT image, and this is then the overlay with the PET. Then another study that was done by the group in Groningen. They labeled trastuzumab, antibody targeting HER2, and used for breast cancer patients. And what they did in this study is that they found out that the antibody dose should be at least 50 milligrams, because if you go lower, the antibody was mainly bound to HER2, which was shed in the blood. And this was going to the colon. So you had a very fast blood PK, and then the antibody is not available anymore for tumor targeting. With this tracer, they could detect most of the known lesions, but for example, liver lesions were sometimes missed, so 3 out of 7. And that's also a little bit of the problem right now, that there's quite some high background of the tracer in the liver, and then liver metastases are not easily found. What was a very interesting finding at that time, but now, of course, more and more often seen, is that with the tracer, with the antibody, a brain metastasis could be visualized. Of course, it's the question, is the blood-brain barrier disrupted in these type of patients if it's a metastatic brain tumor? And that's, I think, clearly shown here, because we see uptake of the tracer in the brain. Then next to this first paper, they also had a case report where they looked also at if you give a higher dose than this 50 milligram, do we also get saturation of the tumor lesions? And that's what they've shown with 280 milligram of trastuzumab. They saw a saturation of 47% of the tumor lesions. So important to think about the dose of your antibody. This is totally different, for example, from small molecules, where you always want a very high molar activity or specific activity. Here, sometimes this is just not beneficial. If you go with a low dose of antibody, it will immediately bind to, for example, in this case, HER2 in the blood, and be cleared, and no antibody is available anymore for tumor targeting. Another antibody that was labeled in the early days is bevacizumab, targeting VEGF. This antigen is associated with breast cancer and lung cancer. With this antibody, the dose shouldn't be too high. So already with 5 milligrams of antibody, good images were obtained. With this antibody, it was actually not anticipated that we could see any tumor, because it's the VEGF is not really inside the tumor, but more in the stroma. What the colleagues in Groningen also showed is that there's significant correlation between the antigen expression and the antibody uptake. that the VEGF is hardly expressed on healthy tissue, so that's why the 5 milligram dose was sufficient. And they also did a study with an HSP90 inhibitor, but there was no effect measurable with zirconium bevacizumab. They also saw a positive trend between tumor uptake and progression-free survival. In this case, there was a combination therapy of carboplatin, paclitaxel, and bevacizumab. The limitation is that there was no distinction between effect of chemo and bevacizumab. Another study, so this was all work mostly from Groningen. This is a study we did ourselves together with the people here at the pediatrics clinic. And this was a study in children, which is quite controversial, as you can imagine. These patients have a pontine glioma. Typically, their prospect is only a few months. So that's why we could do this clinical trial. And what we saw is that in 4 out of the 5 patients, there was uptake of the tracer in the brain. And this was also in line with the MRI contrast. So the blood-brain barrier was disrupted, and then the drug could reach the target. And then in 4 out of the 5 patients, there was VEGF expression. Very controversial, but again, with the total body PET, this could be avenues where we can lower the dose and still get good images. The last example of an antibody that was already approved, that's with all these antibodies, is on a CD20 antibody, rituximab. And there was a study done together with the group in Brussels, and they looked at B-cell depletion and also if they need to give a cold dose before the tracer. They saw that probably really have to look at the amount of B cells. So test that on beforehand to be sure what should be your dose during imaging. And imaging at a therapeutic dose probably is not optimal because you really also saturate then the target, like was the example of trastuzumab. That were the first trials, all with approved antibodies. And what did we learn? Is that we have to be aware of false negatives. The antibody dose during imaging may be lower than during treatment. You have to take into account antigen sinks and also evaluate different dose levels. And of course, there's the non-antigen mediated tumor targeting, so the EPR. And this can also, you can think that the target is expressed there, but it can just be extravasation of your antibody due to the size. After this first trials, we focused more and more phase 1 clinical trials where we applied imaging. And I have a few examples that I would like to discuss. And let's see how far we can get in a few more minutes. So the examples that I would definitely like to discuss is CD44 and PD1, PDL1. First, the study, this was done together with Roche. And we did an antibody, we had an antibody against CD44. And we did mice studies, but the antibody is not cross-reactive. So we had no idea what would be the dose that we need in patients to saturate CD44, which is almost everywhere in the human body. So we did a monkey study. The antibody is cross-reactive with monkey CD44. And we wanted to look at, can we increase the cold antibody dose and then saturate the sinks, so where there's a lot of CD44 expression in healthy organs. And that's what we did here. So you can see on the left, day 2 and day 5 imaging, and then from left to right with an increasing dose of cold antibody. And what you hopefully can recognize is that there's a decrease in uptake in the spleen, bone marrow, and salivary glands. And this is all related to CD44 expression. Then we did a clinical study. dose escalation study. And here you see some data of the biodistribution on the right, and on the left, the PK in the blood. And what you can see, if you have a very low dose, it's immediately cleared from the blood. And this is because there's so much CD44 in healthy tissue and the antibody is bound there immediately. And if you increase the dose at a certain moment, you don't see a difference anymore in the pattern of the curve. So that means you start to saturate. But how does that correlate then to tumor targeting? In this case, we saw that we needed 450 milligrams of cold antibody before we could get any tumor targeting. So that's a really, I think, big sink, to be honest. And typically, this is much lower. And here you see on the top the quite noisy images with the zirconium tracer, and then at the bottom the FDG scan. And you can clearly see that in the head and neck area, there's a lot of uptake of zirconium. So without this imaging, you would have never have an idea what would be the dose that you need for targeting. And also this can help you in optimizing your treatment schedule. The second example is about PD-1 and PD-L1 imaging. And we wanted to see, do we see differences with these tracers? The rationale is that the response with nivolumab is seen across all different types of PD-L1 expression levels. And a single biopsy cannot reflect the heterogeneity in expression within and between tumors. So we had two tracers. The first one is an adnectin, a small protein of around 10 kilodalton, and it's labeled with fluorine 18. And the second one is zirconium nivolumab. So that's targeting PD-1. And we wanted to quantify the uptake and assess the uptake heterogeneity, and also try to correlate the tracer uptake with tumor and stromal immunohistochemistry and also treatment outcome. So here at the bottom, you can see what was done. So you had an FDG, sorry, a fluorine 18 PD-L1 scan, followed by a zirconium nivolumab scan, biopsies if possible, nivolumab treatment, and then the patients were followed until disease progression or unacceptable toxicity. And here you can see then the uptake of the two tracers between the patients, and then also different lesions in the patients. And you can see that it's a very heterogeneous uptake between patients, also among tumors within the patient. And also is indicated then the smaller and the bigger lesions. Then here two examples, a patient on the left with variable PD-1 expression, and on the right low PD-1 expression. And also the different tumors and healthy organs depicted. So if we go from left to right, we start with the FDG scan, a clear uptake in the tumors. Then the PD-L1 tracer, again, good uptake in the tumors, but also a lot in the spleen and the kidneys. And then the PD-1 tracer, zirconium nivolumab, again, a good tumor uptake, a larger liver uptake, and also spleen. And you see also some in the colon. And then on the right, the low PD-1 expression, where you see with the FDG uptake a spot. And again, the PD-L1 tracer, almost no uptake in the tumor. The same for the nivolumab. And again, the same pattern in the healthy organs as with the variable PD-1 expression. Then quantification and correlation with immunohistochemistry. What they saw is if there's no PD-1 expression, the SUV peak of zirconium nivolumab was quite low and much higher if there was a PD-1 expression. And the same was true for the PD-L1. So that's nice to see that there's a correlation. And then with regard to the responding and non-responding patients, that could be differentiated also based on these PET scans, both for the zirconium nivolumab as well as the BMS tracer, so the PD-L1. With the other PD-L1 and PD-1 antibodies, this is not so clear as with this nivolumab. Why this is, I have no idea. This is recently published by our groups and also from Groningen. But this is, I think, a very nice example. It could be that the antibody dose is not optimal in the other trials. We don't know. Few of the time, I would like to show you two last slides. As already mentioned, there are newer developments in the field. Because of the little bit of bone uptake in preclinical models with zirconium DFO, we developed together with Tom Mint a next generation chelator. And it's actually what you see here in black is DFO. Then we call the new chelator DFO star, and it's actually one more unit, the same as in DFO. Then the same linker as in DFO, isothiocyanate, and then coupled to an antibody. And we had a bone metastasis model where we tested trastuzumab. This bone metastasis model consists of HER2 positive tumor cells, and we compared the different chelators and then also a negative control antibody. And what we actually expect is that that in the affected knee, we want to see uptake, and in a non-affected knee, of course, we don't want to see anything. And only in the case of DFO star, that's here on the second image. So this is this first one, DFO trastuzumab. So in the affected knee, you see something, but also in a non-affected knee. So false positive and a true positive. Then here you have a true positive with DFO star trastuzumab and a true negative. This is then the B12. So in both knees, you have a false positive because there's nothing that it can target to. And then with DFO star, you see again with the B12, there's a true negative and a true negative. And the next step is now to bring this to the clinic and see also there if, for example, bone metastasis can be better detected with this new chelator. And the last thing, I think a very nice development for zirconium immunoPET is the total body PET scanner. This is a collaboration with Genentech, where zirconium labeled antibodies were made in collaboration with them. And then in the total body PET scanner, the animals were scanned up to 30 days post-injection. And this is what you see here on a 10-minute scan, day 0, the images look absolutely great. And even after 30 days, you can still see radioactivity, which we typically say it's almost decayed. So in conclusion, I hope I could give you an idea about what you can do with zirconium immunoPETs in the established drugs, but also newer drugs. And there are a lot of studies performed to determine the tumor uptake, but also therapy response prediction. And I think especially what you see now, that it's added to phase 1 clinical trials to get more information in lesser patients at an earlier stage. We think that the new chelator is very promising, and we hope that we can show added value in the clinic. It's debated right now if there's really bone uptake or not. And especially the total body PET cameras, I think, are very important for this field. With this, I would like to thank you all for your attention, and happy to answer any questions, and all of my collaborators and colleagues.
Thank you, Daniëlle, for the nice speech. I don't know if someone has some question. So I have some question. Maybe after they will have other question. I have 2 or 3 questions regarding your experience. I see that we have to inject col antibody to be sure to well target the tumor, and for all the antibodies we have to inject col antibodies before the injection of the radio tracer. And do you have a scale to know which is the amount of col antibody we have to inject?
Yeah, what we typically now look at is the cold PK data that is already available. So if you see a very fast decline, then you know that this is not the optimal dose for imaging. So as soon as you see that the PK doesn't change anymore due to the dose, this is the area where you can start imaging. Okay. So that's what we take now as the how to estimate this based on the cold PK that is known already. If you don't know this, I would say that you have to do a dose escalation to see what is the optimal dose for imaging.
Okay, and how many days before the injection of the radio tracer you inject the cold antibody?
Just before, so maximum 2 hours in advance.
Okay, 2 hours.
Yeah,
okay.
And we have also seen, but it's not published, that, for example, if you first give a little bit of the tracer, we did a very small amount with cetuximab, and immediately after we gave, so that immediately goes to the liver because there's a lot of EGFR. Then we gave cold dose, and then the tracer was going back to the blood. So that's also was quite interesting to see. So in this first few hours, it's quite reversible. And that's why we think we can also give the cold dose shortly before the tracer. Some other groups just mix it, but that's pharmaceutically not always easy because it's a new handling. And in the Netherlands, at least, they don't like that. And then also if you give the cold, for example, quite high dose you have to give, that takes several hours. You don't want to have the radio tracer injected over, let's say, 6 hours, preferably. So then you can do this small bit of radio labeled in a shorter time frame after the cold has been given in the normal. Here we have a phase 1 unit where we cannot have radioactivity. So then they get that there, the biggest dose, and then only the radio tracer afterwards. For some studies where it's only 5 milligram that they get, then we put it in the formulation of the radio tracer. So only if we talk about, let's say, bigger than 50 milligram, then we dose it separately.
Okay, perfect. Because I have in mind that we have to inject the cold antibodies 3 days before.
No.
Yes. No. And regarding all the clinical studies that have been performed with the immunopet with zirconium, where do you see the clinical use of immunopet with zirconium to predict the response, to test or assess the different tools in clinical studies, but at early phase? For you, in your experience, where is the real?
Yeah. The real need is, I think, in early phase clinical trials, where a new drug, where you have no idea what, if there could be, for example, potential side effects or toxicity, that you get more information in this phase 1 clinical trials. We have had a few studies where we really was like de-risking the whole study. So we had a study in patients with it was a liver disease, and the antibody should target, I think it was the liver. It was just not doing at all. So in 3 patients, we injected the tracer, and it was totally not what the company expected. And the whole drug program was stopped based on the few scans. And based on the phase 1 trial, they could not say anything. So it's only about safety, not about targeting. And so on. So we really more and more call it de-risking your antibody development program. With regard to the trastuzumab, there has been a very nice study done with several centers in the Netherlands and also Belgium, the SoFRa trial. And there, I think it's also more like it can be combined, for example, with FTG, where you can really predict which patients will profit from TDM1 therapy, so an ADC. So there it can also be used as an imaging agent, really, and not, really for prediction of your treatment, if the patient will respond or not. But mostly we see the added value in the drug development program, especially because of the high dose right now. If we now can decrease the amount of radioactivity, then potentially we can also look at later time points after imaging. And but drug development, I think, is the most important.
Okay, thank you very much. And after I have a question of the chelator, but you answered to all of my questions, so perfect. I don't know if other colleagues have some question for you, but your speech was very clear, so I think you answered to a lot of questions. so no, so we are on time. So again, thank you very much for your reading and your presence. It was an honor for us to have you. So thank you.
Excuse me, I might have one question. Excuse me, Philippe Garrigue from Marseille. Thank you very much, Daniëlle, for your very inspiring talk and presentation. I was wondering if you had any experience in Amsterdam with nanomaterials or nanoparticle radiolabeling.
Yeah, we did one clinical study with a Dutch small company. It's called Crystal Therapeutics, and it's a nanoparticle, and it has docetaxel inside the nanoparticle. So it's like a targeted drug. No, it's not even targeted drug delivery, let's say a slow release system. And they wanted to see, although it's not a targeted nanoparticle, they wanted to see if they can see tumor uptake with this nanoparticle. And then locally you would get release of docetaxel. And that's what we have shown with imaging. And that's mainly because this study was done because the investors wanted to see at the early phase clinical trials, what is indeed the added value of such a system. Of course, they can measure those docetaxel levels in the blood. If they take, they cannot take a biopsy, so that's more difficult for the different organs. But based on the imaging, and we did preclinical studies and then clinical studies. And we showed that in the beginning, the PK was the same for the docetaxel as the zirconium. And then it slowly started to differentiate, and there was less docetaxel. So it was really releasing out of this compound. And that was done by the PK, and then we can also show tumor targeting by the zirconium SNCL. So yes, it has been done, and this was just a non-targeted nanoparticle, but it was very valuable information for them to know. And also very valuable information, we didn't realize that afterwards, only afterwards, is about nanoparticle synthesis. Because in the production process from a small scale to a large scale, we faced some issues, and we didn't expect that this, we could really see with imaging what the problem was actually. And this is something quite neglected in these things, in these nanoparticles synthesis. But yeah, as soon as the paper is out, I can tell the whole story, but it's under review right now. But the imaging was really helpful for this company.
Okay, thank you very much.
Yeah.
We have another question for a shy colleague. He preferred to write. It is a technical question. Is labeling with zirconium possible with basic equipment available in an hospital pharmacy like the mini IO automat or something like that?
We actually do everything by hand. So not even automated, because the amount of radioactivity that you use is so little. So we only start with, let's say, 70 megabecquerels or maybe 80 to make a dose of 37 megabecquerels. So in that sense, and we do everything by pipetting, and that's all doable. Doesn't give a lot of radiation dose compared to, for example, fluorine-18 tracers, where you need a lot more to start with. So in principle, if you have the area is suited to make radiopharmaceuticals, I would say it's possible.
Yes, I know that Tracis with Mini IO developed a cassette and something to make radio labeling with zirconium.
Okay, yeah, we also used, I have to recall the name of the company. We also automated the radio labeling, but especially purification is not that easy, I have to say. That's the main obstacle, purification.
Okay, so perfect. I don't know if someone else wants to ask something, but thank you for your time.
You're welcome.
And see you next time, I hope.
Yeah. Okay.
Bye-bye.
Have a nice meeting. Bye-bye.
Bye.
Et Charlotte, je ne sais pas si on peut poursuivre la discussion, bon, du coup en français vu qu'elle est partie, sur peut-être l'accessibilité du zirconium au niveau national en France et voilà, sur votre expérience à vous, ou peut-être l'expérience de différents centres parmi les différents participants connectés.
Alors, je sais qu'il y a un essai, enfin, plusieurs essais cliniques qui ont commencé. Donc, effectivement, la Hollande était les premiers avec, comme l'expliquait, les Belges. Maintenant, c'est arrivé en France. Je sais que Fred, Bordeaux est inclus dans un essai clinique ainsi que Nantes. Alors, je ne sais pas, peut-être que vous voulez en parler un peu plus.
Effectivement, oui. Mais alors, pour le moment, donc, on bénéficie de médicaments prêts à l'emploi. Donc ça simplifie grandement les choses, effectivement, avec anticorps radiomarqués, donc pour tout ce qui est pathologie rénale, et du coup avec une injection, par exemple le vendredi, et un passage en caméra le mercredi de la semaine suivante. Mais globalement, effectivement, après, en termes de pratique de radiomarquage, les choses sont un peu plus éloignées dans le temps, hein, clairement. Enfin, nous, en termes l'aspect pratico-pratique sur cet aspect-là, enfin, c'est pas pour de suite. Il y a des contraintes de coût qui sont aussi assez conséquentes, et puis d'accessibilité, on va dire, à la matière première de manière générale. Après, sur tout ce qui est particules ou autre, effectivement, alors je ne sais pas si Alain est dans le coin, mais parce que, on va dire, à l'ouest, enfin, le nord-ouest a une grosse expérience sur le développement et ses différents marquages. Je sais qu'également à Nancy, il y avait des perspectives de travail sur ce type de radionucléides. Et à Marseille, vous en êtes où, Philippe? Vous avez des velléités aussi vis-à-vis de ça?
Non, non, pour l'instant, non. Je sais qu'il y avait peut-être Strasbourg qui, qui avait aussi l'intention de se lancer sur le zirconium. Je vois que Nicolas a allumé son micro pour Nancy.
Alors, après, Strasbourg, le fabricant, le Zirconium, ils sont capables de pouvoir approvisionner, hein.
Voilà.
Ouais.
Oui, il y a 2 sources d'approvisionnement actuellement. Ça marche plutôt bien. C'est toujours aussi cher, mais ça marche plutôt bien.
C'est-à-dire, quand tu parles de source d'approvisionnement, c'est quoi? C'est-à-dire, c'est le cyclotron qui est sur place ou tu fais livrer du
Ah non,
zirconium
tu fais livrer.
comme
Tu fais
précurseur?
livrer. Tu peux faire livrer, hein, c'est pas un souci. Tu payes ça en nombre de millicuries. Tu planifies ton jour de livraison. Ils ont un planning. Il n'y a pas de grosses difficultés là-dessus. Et après, c'est la recette de cuisine est sur Internet. Tu as même une vidéo sur je ne sais plus quel site qui explique ça très bien avec la méthode de contrôle qualité, la liste des réactifs. Enfin, c'est le vrai guide du petit chimiste. Et c'est suffisant pour se lancer.
Et c'est Perkin-Elmer, comme elle a dit, qui vend ça?
Ouais, tu as Perkin-Elmer et puis tu as quelques productions aussi sur Circé, le gros cyclotron qui est à Cronenbourg.
D'accord. Okay.
Anthony, si tu veux, je peux t'envoyer les coordonnées de Cronenbourg.
Volontiers.
Mm.
Je ne refuse pas le Cronenbourg, pas de souci.
Peu de monde le refuse.
On a une usine Cronenbourg à Marseille. Non, même pas, c'est Heineken, pardon.
Et après, Frédéric, ou ceux qui ont déjà injecté là pour un essai clinique, vous injectez du froid donc avant. Et après, c'était savoir s'il y avait des mesures particulières en termes de radioprotection chez les patients, parce que vous les injectez le vendredi, ils retournent chez eux sans aucun souci, ou est-ce qu'il y a quand même des mesures de précaution qui sont prises ou données?
Modalité de radioprotection, globalement, c'est assez light. J'avoue, c'est assez light. Alors, les quantités ne sont pas non plus énormes. On a aussi des problématiques, même sans aller forcément jusque-là, c'est aussi pour nous en termes de radioprotection, etc. Parce que en général, tu as des gros volumes. Les injections, les modalités d'injection sont assez, pas lentes, mais nous, par exemple, la mise en seringue, on a juste nos moyens de radioprotection pour la mise en seringue, mais c'est pas optimum. Donc, il y a aussi, rien que sur la partie mise en seringue et administration, quelques ajustements à faire. Et après, non, nous, les patients sont libérés. Et voilà, tu as certaines modalités qui sont prévues dans le cadre de l'essai clinique, c'est l'essai zircon avec le girentuximab, mais globalement, non, les choses sont. De toute façon, le discours en termes de radioprotection sur le plan diagnostique a conduit à une certaine simplification, hein. Donc, voilà, à partir du moment où on n'est pas sur la thérapie, dans la plus grande. Alors, il n'y a pas de discours clairement officiel là-dessus, mais voilà, on va vers une diminution des recommandations en termes de radioprotection et ça n'y déroge pas forcément. Du coup, c'est light.
Et après, vous injectez du froid avant, alors, comme elle le disait.
Non.
Non.
Aurore Recherche, du coup, radiopharmacien sur l'ICO, centre anticancéreux à Nantes. Je ne sais pas si vous m'entendez.
Oui,
Ouais.
tout à fait, ouais.
Du coup, nous aussi, on a commencé avant que ça soit transféré sur le CHU, sur l'essai Zircon, et on a 2 autres nouveaux essais avec le même anticorps, un dans le cancer du sein et un dans la vessie. Et effectivement, j'ai été surprise, on n'injecte pas de froid avant, pas du
Ouais,
tout.
nous non plus.
Et de la même manière, je suis assez d'accord, au niveau de la radioprot, c'est assez light. Effectivement, il n'y a que la mise en seringue qu'on fait du coup en enceinte blindée haute énergie. Et après, étant donné qu'il faut faire une injection lente sur 3 minutes, avec un volume de 10, du coup, on utilise un pouce seringue derrière un paravent plombé et c'est tout. Voilà. Mais ça pourrait être intéressant d'avoir des petites recos ou quelque chose par rapport à ça. Et pareil, les patients, ils sont gardés le temps de l'essai parce qu'il y a des prises de constance et une surveillation à faire dans la journée, mais après, ils rentrent chez eux, effectivement. Après, on est sur 37 mégabecquerels, mais bon, voilà.
Après, ce qui serait assez intéressant de faire, c'est parce que du coup, on va avoir pas mal de matériel à tester pour ces différentes injections, etc. Et ce sera aussi tester toutes les interactions potentiellement contenu-contenant qu'on sera susceptible de rencontrer avec toutes ces gammes de produits. Parce que je pense aussi qu'il faudra faire gaffe à ce que l'on utilise, que ce soit pour la mise en seringue, pour l'injection, pour limiter toute perte d'activité entre la mise en seringue et l'administration patient. Je pense qu'il y a quelques trucs à regarder aussi.
C'est un peu hors sujet, mais il y a une autre application qui est intéressante avec le zirconium, c'est le marquage de cellules. C'est faisable de faire du zirconium oxyne qu'on peut utiliser comme on utilisait dans le temps l'indium oxyne pour marquer des cellules.
Ouais, tout à fait.
Dans le temps.
Nous, on fait plus, on fait du techné. Mais...
Non.
Ah, c'est super ça pour les plaquettes, par exemple.
Ouais.
Ça
pourrait être intéressant parce qu'effectivement,
faire des bioséquestrations complètes, super.
Ça peut être intéressant à développer, ouais.
Même les blancs, même les leucocytes d'ailleurs. On aurait des belles images le lendemain. Enfin, plus tard. D'accord.
Anthony, j'envoie dans le chat l'adresse mail de tous les gens là que tu peux. Enfin, c'est surtout Nathalie Dick, tu verras. Mais après, c'est tous les gens qui sont impliqués dans la synthèse. Voilà, je te les ai mis là.
Merci beaucoup.
Bon, merci à tous. Merci pour vos questions et pour les interactions. J'espère que ça vous a plu, surtout, et que vous avez appris des choses sur le Zirco. Et puis, maintenant, c'est Julien, je crois.
Tout à fait. Merci beaucoup, Charlotte.
Avec plaisir.
Vous m'entendez?
Oui, Julien.